And we're on. So I tried to do a couple of different things on this Friday session, and I'm going to just switch over to a PowerPoint slide. So hopefully you guys can see this now. Uh, the Friday sessions are an opportunity for us to go over some ideas that I present slightly differently than perhaps they end up being presented in the book or in some of the videos. Also, an opportunity to do some problem sets together and to get you guys working on some problems that might help with both the homework, but also help in lab. There are a bunch of different people who are working in the laboratory sessions, and there are a variety of different problem sets and ways that people are approaching them. So I try to match up as much as possible to try to get people in sync so that lab is a little bit smoother as well. So we cover things in a little bit more detail. And you'll see some of this overlap, especially as we start talking about, for example, chi-square analysis. Chi-square analysis is a key component of genetic analyses. And so you're going to find you'll be doing a couple of different chi-squares in laboratory, but also we're going to talk about it in class. You'll also notice that I'm finally beginning to figure out the achieve settings in the textbook. Most everyone, I still think there are 3 people who either are in the process of dropping the course or have not figured out how to get the online textbook. If you are having issues, please email me. Let me know, just email me. I get back to you as soon as I possibly can, especially now that the internet is up and working again. And we got to get you online with the textbook. Remember that all of the homeworks, there's a little bit of crossover in terminology in the syllabus because we've changed from having Friday problem sets to basically homework is due on Friday. I had the homework set incorrectly. Some of you noticed that it showed up as being due at 12 a.m. this morning. I did not expect you to get up this morning at 12 a.m. to submit your homework. I think that that's just, that was a mistake on my part. So I changed it over for everyone to 11:59 this evening.
Perfect.
Also, you should be able to access those homeworks throughout the entire course. Someone was saying that they could not re-access the homeworks. I reloaded the sessions and reset some parameters, so you should have access to it throughout the course. If there's some issues, I'm not going to lose too much sleep over it over this week. I think I've reopened them a few different exercises for people already. However, we've got to get on track, and so that's why I'm emphasizing if you're having issues with the Achieve Network or the online textbook, you really got to get those up and going. So Macmillan support has been fairly good, but the quicker you can get to them, the quicker they can help resolve an issue.
Professor,
Yes.
I know that we're in class right now, but before I logged on, I checked the homework and I finished the homework earlier this week, but it didn't register that I completed homeworks 1 and 2. So I don't know if after class we could take a look at it.
Um, yeah, sure. We can pull it right up and see what's going on, or I can check, or just send me an email.
Okay.
It's probably quicker and easier just to let me know. Okay,
All right. Thank you.
no problem. So it's Friday, and I tend to I am a huge music fan. If you could see behind me, besides these people working in my lab, you'd probably see a couple of my guitars hanging on the wall here. And I figure Friday is as good a time as any to start our Fridays uh, with a little bit of music. So, and of course, it should be genetics. And so here is Turn It Up. Hope everybody's awake in your house. So there you go. These we'll see a couple of this guy's videos. He's a friend I met when he was a postdoc in molecular biology at Stanford. So it just goes to show you never know. Extra credit if you know the original, if you know the original song. I don't know what. There we go. No, we don't want to see that again. So, as you know, genetics is designed as a Q2 course. So genetics is all about math. And so I like to, I found this little problem that is not really about genetics, but rather kind of gets to help me understand where the class sits in terms of their quantitative expertise and how their quantitative reasoning skills are. And so I love this problem. It really came about. I like the way it was set up by the movie, or actually the book, The Martian. Anybody watch it? The movie, The Martian? No?
A little bit ago, I saw it. It was pretty good, actually. I really appreciated it.
Yeah, the guy who wrote it is actually a physicist, and he wrote it on a blog originally, and then eventually became a textbook and made into a movie. So he's really a lot of it is very scientifically accurate. It's a fun movie for those of you, if you haven't seen it, I highly recommend it. It's a good movie. But this comes about like this. And so, if you guys got to make sure you got a pen and paper handy, as I mentioned early on, when we're doing the online work, a lot of it, if we were in class, we would all have notebooks. And in fact, ugh, I love these notebooks. These are handy, fading in and out thanks to my background. But I love these composition notebooks for you to keep track of problem sets and homework. It's a great way to keep notes and to keep up with the work in the class. And in fact, if we were in person to person, one of my requirements when I teach the course is actually everybody has to have one. They usually go on sale at Staples, 4 for $1 just before the semester starts. So they're really not that expensive. But this problem can be solved a lot of different ways. And Matt Damon, if you did see it, you recall he grew potatoes on Mars. He became stranded on Mars by himself, all alone, obviously with equipment that was along with him for a scientific mission. And one of the ways he survived until he could be rescued was he figured out how to grow potatoes on Mars. So in our little thought experiment here, we know Matt Damon grew 100 pounds of potatoes. And potatoes, this is not true, by the way, it's just for the problem, are 99% water by weight. So they're 99% water. In order to store them properly, he has to dehydrate them until they're 98% water. Okay, so he's got to dry them out from 99 to 98% of water. After he evaporates that water, how much do his potatoes weigh? So I want you guys to take a couple of minutes and think about this. I will tell you ahead of time that there are a couple of different ways to solve the problem. And so I want you to go ahead and think about it. I am going to actually take a moment here and I'm going to put a little poll. And let's see, where's my poll? And while you're thinking about it, I'm going to put a little poll on, which you should be able to find and see what you think. So any thoughts on how would you approach this? How would you approach the idea? How would you solve the problem? I'm going to go ahead. What's that?
I was going to say, first thing I noticed, of course, is that you have 99% water on the on the potatoes initially, and you have 100 pounds overall. So 99% of that, you have 99 pounds of water,
Right,
1% of solid material.
right, only 1%, exactly correct. So I know you guys are, a lot of you are blacked out. If you don't get involved, I'm just gonna call you out, and you got to kind of get into the thought here. So what are you thinking? How would you, now I know most of you are trying to think it out. You're trying to reason it using your verbal processing. How would you do it mathematically? What do you think? Aman, any thoughts?
I'm not entirely sure how to explain it.
Ah,
It's just sort of I'm doing it in my head. I don't know how to explain it to you.
oh, okay. So you're using verbal reasoning.
Yep.
Okay. Eunice, what do you think?
I'm probably overthinking it. Like he said, I don't really know how to explain it because I also don't want to sound dumb.
Okay, no, you can't sound dumb. It's you can't sound dumb. It's your thought process, and I think everybody has a habit of using verbal reasoning, right? I mean, you're thinking it through in your head.
Yeah.
Okay. So what if you were to write down like some kind of an equation? What would you do mathematically? Let's pretend we're in a mathematics class.
I would do like algebra
Ah,
and do like the water weight minus the water weight, kind of, and then I would get like how much I have left. I would say like dehydration,
okay.
but I don't really know how to go about it.
All right. Well, that's good. That's good thinking. Anybody else? Different way or the same way? Can you add to that?
Potato weight before dehydration, if we're talking about the solid weight of the potato, is not going to change at all. So that's
Ah,
going to have to remain constant.
excellent. Excellent. Right. Because the potato's not disappearing, just the water. Very good. Anybody else? JC, what do you think?
I wasn't too sure, but
That's
I thought the
okay.
same thing where there was like 99 pounds of water with
Uh-huh.
99% of water and 1 pound of potato.
Right.
So I wasn't sure if you had 98%, if it'd be one less pound. But I've never
Okay.
tried to do
So
the math, so it's just a guess.
you're trying to do ratios.
Yeah.
Aha! God, I knew I took all those math courses for some reason. No, I think you guys are right on the money with the process of your thinking. But part of what we've really got to get used to, and you'll see this as more time goes by in this course. And I'm going to go ahead and end the poll. Did everybody pop in? It looks like a couple of people did not, but that's fine. Let's go over a couple of different ways. It looks like everybody's kind of focusing in on they really don't change much. Okay. And again, part of the reason that we arrive at that kind of an answer is because we're using our verbal reasoning skills. All right. Part of what we're asking you to do in genetics and in Q2 courses is not to use your verbal reasoning skills. And that's what makes genetics hard. That's why people have a hard time, because sometimes our verbal reasoning skills are exactly the wrong way to solve a problem. And so let's take a look at how one might go ahead and solve this particular problem. And remember, we haven't gotten to genetics yet. So one way to do it is just to do it exactly as a couple of you guys were saying, which is we know that 100 pounds of potatoes, you get 99 pounds of water and 1 pound of potato. And the question here is, of course, what we want to know is how much is it going to work? How much is the bottom line going to weigh if we have 98% water, which means that there's 2% potatoes? But of course, we know that the amount of potato doesn't change. What changes is the proportion of the potatoes. And if we were to look at it in this context, we could figure out pretty quickly what's 2%, okay? 1 pound out of a total of 50 is 2%. So that the correct answer, believe it or not, ends up being 50 pounds of potatoes. So this is using a combination of just simple mathematics, being able to do percentages, and a little bit of verbal reasoning. So this is one way you could figure it out. Now, I don't know about you guys, but just reading the first time I read the problem, I had no clue that it was going to be so small. I mean, nobody answered on the poll and said, "Oh yeah, it's going to be 50 pounds or less." Everybody was focusing on that 99.98. Human beings are terrible, and I'm going to say this over and over again. Human beings are terrible at intuitively grasping mathematical concepts. We have to be trained to do it. So that's part of the purpose of this course, and hence its Q2 designation. But I said there were multiple ways to solve the problem. So another way you could solve it is using basically ratios. And using ratios is pretty easy. Notice that we're rephrasing the question, so we're still using a little bit of verbal reasoning. And we say 2% of what is 1 pound? And so we use our ratio, 2 out of 100, 2%. We want to know what times what equals 1 pound. And of course, if we solve for X very quickly, we can show that after dehydration, we have 50 pounds. So we get the same answer using a slightly different methodology. Of course, there's even another way to do it, which is actually using algebra, because what we really know is how much water is there when we start the experiment. We know it's 99% out of 100. How much water after we dehydrate? It's 0.98 times 100 minus X. And in this case, X is what its future weight will be. And of course, if we subtract the weight of water minus the water after dehydration, we actually get X. And you can solve for X here, and everybody knows, right? 0.99 times 100 would be 99. And then, of course, so let's do it up here. We know that we've got 99 pounds minus 98 minus X. And then, 0.98. You guys are supposed to correct me when I make mistakes, you know. It's hard to do this in real life. Wait a minute, let me get rid of this. Right? Because 0.98 times 100 is 98. And then we're subtracting 0.98x equals x. In order to solve for this, we have to do 99 minus 98 is 1, minus 0.98x equals x. We can then add, we then have to solve for in here. And of course, if you solve out using simple algebra, it still equals 50. So really pretty straightforward algebraic approach. So you could see how you can use 3 different methodologies to get at the same answer. However, each of the methodologies required some mathematical expertise writing down the equations. And so that's what I really want to encourage you guys to do, is to pay attention to the question being asked and find a way to express that, meaning writing it on paper, not just thinking about it in your head, but writing it down on paper so you could eventually get to the point where you can actually have an equation describing the situation. So let's take a look at applying some of these ideas to genetics. This is a drawing by Sir Reginald Punnett. This is Punnett of Punnett squares. And in fact, this is the original Punnett square. This was from a paper that Punnett wrote in the Proceedings of the Royal Society of Medicine in 1908. It is actually his drawing. Back in the day, there were no computers to make drawings in published papers, and this is what he did. And this is where our original notation comes into play. Notice that he has up top, he has P. He shows that gametes are made. There is now an F1 cross, gametes of F1, and the F2 cross. Each of these has very, very specific definition. P, of course, obviously can stand for parents, but also from a genetic sense, that's P, pure lines. And the reason we define pure lines is that here we've got a black and here we've got a white, and they are pure line. That is, when these individuals are bred within themselves, they always produce progeny of the same phenotype. So that is, if you made a bunch of these black individuals together, all of the progeny will be black, and you never ever see a white one, or at least extremely rarely. Okay. And so that's why they're called pure line. We actually use the term pure line before we can use the term homozygous. Oops. There's an S. Before we use the term homozygous. And the reason is fairly simple. Before we do crosses, we can't determine what the mode of inheritance is. I'm sure you all recall from high school biology, from Bio 1, Bio 2, whatever, we know that all organisms are diploid. That is, there are 2 alleles at each genetic locus. Everybody know what a locus is?
The spot on the chromosome where the gene shows.
Exactly. It is a location, right? Locus, location, same root word, same root word. And in fact, we know that there are 2 copies of each gene because we have 2 chromosomes from each parent. And so we know homozygous looks something like this, or like this, or like that. There are all sorts of ways to define it, but the allele at each locus is in fact identical. But we can't define whether it's a single gene, multi-gene, or perhaps not even a gene. We can't define that until we begin to do crosses. And that's where Punnett really took Mendel's work and really expanded it. He recognized, because of Mendel, the role of gametes, and that, of course, nobody knew anything about the names gene or locus. A particle was created in the terms of gametes, so we know meiosis occurred. And these gametes, if we took a pure line black and a pure line white and mated them together, we know that the gametes would get one copy from black, one copy from white. So therefore the F1s would be half and half. What we now know, and of course, we say they are heterozygous, and in our notation, looks something like that. That's a small a. Okay. And F, of course, stands for the word filial. Comes from the Latin. Some of you may be more familiar with the French, fille or jeune fille, son or daughter. So filial are the sons and daughters of the previous mated generation. So we see in the first filial generation, all the individuals are heterozygous. And then here comes the fun part. When these F1 individuals that are heterozygous undergo meiosis, half of the gametes will contain a black allele and half white. Same thing on the other side, half black, half white. And there are 4 different ways these can combine. We can get the female black allele with the male black allele, female black with male white, female white with male black, female white with male white. And of course, these individuals are now, what do we call that? What do we call an individual that has genes from each of their parents? They have 2 copies. I'll give you a clue. We all have 2 copies of a gene at any locus. Got it now?
Diploid?
Diploid, absolutely. And if we know that these diploid individuals have 2 copies, what's the term we use for gametes?
Haploid.
Haploid, absolutely. Good. Okay, so pretty easy. And of course, you could see here, we have the famous Mendelian ratios. We have 1 homozygous black, 2 heterozygous black and white, and 1 homozygous white. This, by the way, was the reason that it was called a Punnett square. Punnett never drew a Punnett square like we all have all been drawing them since 7th grade biology, maybe even before that, depending on your school system. He never had that, never did it like that. Punnett always drew it exactly like this, showing gametes and then the F2 individuals in the middle. So really kind of a nice way to look at it. So I like this. I love this drawing because it really focuses on The important thing to remember from the video, even if you don't remember the original song, the really important thing is meiosis, is the process of meiosis. And we'll be talking about meiosis over and over and over again. But all of the nomenclature here is extremely important. So I want to give you another problem. And this problem is actually the most important problem that we're going to do all semester. We are going to come back to the idea expressed in this problem over and over and over again. Okay? And it really has to do with a very, very simple phenotype: coat color in guinea pigs. Guinea pigs, if you recall, were actually domesticated for their meat production in South America. These particular crosses were actually done by a geneticist, Sewell Wright. Sewell Wright, while he was at the University of Chicago. The University of Chicago did not have animal care space. So unlike Stockton, where we can go down and visit our animal colony and see all sorts of different beasties, we will see some of them in genetics lab a little bit later on in the semester, probably in about 3 weeks or so. So Sewell Wright had a guinea pig colony in the basement of his apartment building in Chicago. So this is a real committed geneticist, a committed professor, where he maintained all of his experimental animals in the basement of his house. Actually, it was an apartment building. So in this case, we were looking at the trait of black and white hair color. Hair color is also called pelage, another vocabulary term. It's body hair color, okay? Body hair color. And black is dominant to white. So black coat hair can be either homozygous cap B or could be heterozygous cap B small b. And an important part of genetic notation can be seen within these parentheses here. And you could see this underline. What that underline means is we do not know what the second allele is. We know that there's a cap B because I've already told you that black is dominant to white, but a guinea pig could be black because it's homozygous cap B or because it's cap B small b and therefore heterozygous. If you don't know or it doesn't matter what the second allele is, you use an underline. Okay, white obviously is always heterozygous, small b, small b. Here's the question. So get your little pen and paper out. If we have a pure line black guinea pig, what does pure line mean?
Homozygous dominant.
There you go, homozygous dominant, absolutely. If a pure line black guinea pig is crossed to a white guinea pig, what fraction of the black F2 is expected to be heterozygous? Now, I want you to think about it for a minute. I'm going to put another poll up for you. These polls are so much fun. If I can find it. Ah, there we go. Oops, sorry. Don't panic. There we go. I always make these polls anonymous, just so you know. I have no idea where my poll just went. Crud. All right, so my poll died.
You really meant it when you said no one answered 50 pounds.
Yeah, sorry. I thought it automatically shared the results with you. I guess it doesn't. Zoom drives me crazy. Yeah, nobody guessed 50 pounds. Here we go. We'll stop sharing. That's embarrassing. Wow. Oh, wait a minute. Here we go. There it is. Now I get it. Okay, so we're going to launch the poll. So everybody got a chance to think about it. I highly recommend, if you're still thinking about it. So here you go. There's the poll. If you are thinking about it, what can you do? Remember, we're talking about not just thinking, but actually writing these things out. So, right? So what are we going to do? We're going to do a Punnett square. If we look at the male, we know that if a pure line black, so what's the genotype of the pure line black?
BB, capital both.
Cap B, Cap B. How about white?
Lowercase bb.
Okay. So what's that mean? We just fill in all these guys. Now, let's read the question. What was the question? Is that an F2 cross right there?
It says F1.
Yes, that is an F1 cross rather. We need the F2. So notice that the question, and this is absolutely typical of genetics questions, the question assumes that you know to make the F1 cross, which means all of the individuals, just as we saw in the previous slide, are heterozygous. So it means our Punnett square looks something like this, right? Now, I'm going to end the poll. And now that I know to share results, what do you think?
I mistakenly did not pay attention to the F2.
There you go. Good. So that's really important to know. And in fact, this confuses students. Heck, it confused me the first time I did it. I'm like, why would you not ask me about the F1? And the answer is, in all of the genetics problems that have ever been done, they just immediately assume they tell you the inheritance pattern, and then they assume you're going to know to do the F1 cross, and then go ahead and do the F2 cross.
I have one question about this though.
Yeah.
So in F1, of course, the generation is going to show 100% heterozygous pattern.
Exactly.
But in the F2 generation, is it assuming that the same offspring from the parent generation are being inbred?
Yeah, pretty much.
Okay, yeah, because that threw me off a little bit too.
Yeah, pretty much. If you're thinking inbreeding, you're like 2 months, a month down the road.
Okay.
So you're absolutely right. From a practical point of view, that's correct. But for our purposes, we just kind of assume that these are individual crosses and we don't really care that they're all related. So what about the answer? So let's assume, let's go ahead and say, okay, so now let's read the question again. I'm going to slide my, I don't know if that moves your poll result or how you see it. So what do you think about the answer? Now that we've got our Punnett square, the
It's gonna have to
question
be
is
1/2 or 50%.
50%. How about everybody else? You want me to redo the poll?
Wouldn't it be
Percent too.
2/3?
What's that?
Wouldn't it be 2/3?
2/3? How'd you get 2/3?
Well, it's out of black, so only 3 of them are black.
Ah. Oh man, whoever wrote that poll made a mistake. We know that the homozygous small b is white. So when we ask the question, what fraction of the black F2 is expected to be heterozygous, we're really only talking about these 3, and we have 2 out of 3 total. So the correct answer, sorry, that was a little tricky not putting the correct answer in the poll. The correct answer, of course, is 2/3. This is without a doubt the most important problem that we're going to do. There is absolutely no question. We are going to see this question over and over again when we do pedigree analysis, when we start talking about genetic testing later on in the semester, because what happens in genetic testing or pedigree information? We're trying to identify the genotype of an individual of known phenotype. So if, for example, a couple comes into your office as a genetic counselor or as perhaps an OBGYN or a general, a GP or a family physician, and a couple comes in and says, hey, neither of us has a particular disease, but our parents do, or perhaps our brothers and sisters do, or perhaps our brothers and sisters had children with a disease. And we want to know what are the chances of our children either having the disease or being heterozygous. Notice I didn't say carrier. I hate that word. No such thing as a carrier. You didn't pick up a bad gene or a deleterious gene on purpose and carry it around just to give it to your kids. No. All right. You're heterozygous. So what are the chances of that happening? Well, your chances are skewed because we already know what your phenotype is. So this problem is extremely important all the way down the line. We're going to see it over and over again, at least by my count, at least 3 more times during the semester. My suggestion at all times is, for now, always draw your Punnett squares. I know it sounds a little overly simplistic, but draw your Punnett squares. It will help you tremendously. It will help you tremendously. I do want to do one thing before we finish up, and I'm going to turn this over into a whiteboard here, if I can figure out how to do it. Don't panic. There we go. There we go. So we have a whiteboard. And what we really did in the previous example was something like this. We took a pure line, crossed it with another pure line, created an F1 cross. So here are our P1s, F1 cross, and then of course, got to an F2. However, rarely do we spend a tremendous amount of time working on such a simple cross. The next most complicated one, of course, that we do, and this will be, those of you will either approach it, either be doing it this week or next week in genetics lab, is a dihybrid cross. So again, dihybrid. What it means is instead of one gene, which would be a monohybrid cross, that was our black and white guinea pig example, a dihybrid cross actually has two genes. And in this case, we've got the double homozygous dominant and the double homozygous recessive. And so the dihybrid cross is, again, one of the classic examples. And you may, of course, recall this. Some of you, this is old hat. Some of you, this is fairly new, but I want to take the opportunity to go over it. So we have a pure line. So what do we know about gamete production? What kind of gametes can our double homozygous dominant individual produce? Well, we know in the previous example, we could get, if we were only dealing with a single locus, we could have A. In this case, we have a second gene. So what gametes at this locus can be produced? Right? Which means, and you just count them up, A, B, A, B, A, B, A, B. Okay, so this is just using a little path diagram. It seems silly to do this for just two genes. Another way to think about it, we can perform meiosis by going like this. You get a small a, small b, small a, small b, small a, small b, small a, small b. So we have 100% A, B gametes. All right. There are a variety of ways to do this. You do what's most comfortable. I love this little path diagram here. And the reason is it is extremely simple when it comes to only two genes. But what is really difficult is all of a sudden, if we were to add another gene, right? So now all of a sudden, we can really use these path diagrams. The other thing that's really nice about using these path diagrams is it converts very nicely to a computer programming model in bioinformatics. So a little bit later on, if you continue in genetics, this saves you a lot of grief by learning how to use these path diagrams. So we know that our F1 population will always be a cap AB gamete combining with a small ab, which means that all of our F1 population are double heterozygotes. double heterozygotes. What's this mean in terms of our F2 population, where we're actually crossing 2 double heterozygotes? So let's think about it. What gametes can each of these heterozygotes produce? So anybody have a sense? I'll go on. Don't make me call on people. Rose, what do you think?
Could you repeat the question?
What are the gametes that are produced by these heterozygous individuals?
Oh wait.
So again, we can use our little diagram like this. Make life easy. Which means that, what do they have? Because you have to have every locus or every gene has to be represented in the gametes. Okay? So what are our combinations? I'll get you started. AB, cap A, cap B. Now what?
Capital A, lowercase b.
There you go. Well, now what?
Lowercase
Lowercase a, capital B.
Good. And finally,
Lowercase a, lowercase b.
oh, so easy. See that? Once you get the hang of it. And then, of course, in our, those are female gametes, our male gametes, again, will be the same because they're both heterozygotes, which means that what kind of loci are these? If they're the same in both males and females? Ooh, what a great question. Well, if they were on the sex chromosome, what would they be?
These are autosomal, right?
Autosomal. Excellent. They are autosomes, right? And then, of course, so we've got small a, cap B, small a, small b. And of course, we now fill this all in. Oh my gosh. All right. How fast can you go? I never leave myself enough room. Keep going, keep going, keep going. Don't stop. Whew. Oh, my hand's starting to get tired. Notice you always put the capital or dominant letter first, which is really not the best way to look at this because it obscures the parent that particular allele came from. But we'll talk about that a little bit later. You guys are checking me for mistakes, right? I don't know. Starting to lose track. Whew. Wow. One of the fun things of looking at these matrices, I'm going to change the color of my pen here. See if that worked. Ooh, it did. Okay. One of the fun things of looking at this, first of all, you should know that in every F2 dihybrid cross, how many total progeny are expected on average? It's a 4x4, right? So you get 16. And you can actually see patterns begin to develop here, okay? Look at the diagonal. Notice anything about that diagonal?
They're all the same.
Yeah, they're all the same. Not only are they the same, every one is a double heterozygote. A double heterozygote. How about, let's look at another one. Okay. Double homozygote, double homozygote. How about homozygous cap A, heterozygous cap B, homozygous small a, heterozygous B. How about, I think I'm running out of colors, don't panic. Heterozygous A, homozygous cap B, heterozygous A, homozygous small b. See if I can get one more color here. Homozygous cap A, homozygous small b, homozygous small a, homozygous cap B. So genotypically, we end up with 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. So genotypically 1, oops, sorry about that. I'll go back to black. 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. So we see these patterns developing. We'll talk a little bit later on. This pattern should look very, very familiar to you. Very familiar to you. We'll talk about why a little bit later on. Importantly, so this would be your genotypic array. But what's more common is the phenotypic array, which what are or commonly known as Mendel's F2 phenotypic ratios, which you should remember as what? What are the phenotypic ratios expected from an F2 dihybrid cross? Ooh. Well, the F2 ratios on a monohybrid cross are what? 3 to 1, right? So how about a dihybrid cross? Oh, I know some of you know this. Does the number 9 ring a bell?
9 to 3 to 3 to 1.
Yeah, piece of cake, right? And weirdly enough, what if we add these up? 16, so you know you're right. The key element here is when we talk about what phenotypes are expected. Sorry, that's a 9. My pen is kind of goofy here. The 9 are 9 of the 16, 9/16 of the individuals will express both dominant phenotypes. 3 will be dominant of one phenotype and recessive of the other. 3 will be recessive of one phenotype, dominant of the other. And only 1/16 will express both recessive phenotypes. So it's a good way to think about the F2 phenotypic array. When you think about 9 to 3 to 3 to 1, in your mind, what you want to remind yourself, it's 9 double dominant, 3 dominant and recessive, 3 recessive and dominant, and only 1/16 will be the double recessive phenotype. Okay? And I think that's a great place to stop for today. I am going to stop that share. Hopefully it remembers to save it for me. In any case, this will be online for you guys as soon as it downloads and converts. I hope that made some things a little bit clearer for some of you who may have been a little confused up until now, and walking through it was a little bit of an adventure. Hope you like the video. I got a lot of those. They're fun. They're fun. Any questions?
I have one question.
Yep.
So we're going to be definitely focusing on a lot more of those larger Punnett square problems in this class.
What do you mean by larger? You mean like more genes?
More genes, yeah, more alleles.
Not maybe we'll go to a maybe a tri-hybrid once, but I wouldn't get more complicated, because you get to the point where you need a computer in order to be able to work them out. I mean, think about it like this. There are, and you probably read this in the textbook by now, even though we're not talking about the biochemistry of the gene just yet, there are approximately 20,000 human genes.
Yeah.
So now imagine doing a Punnett square with crossing 2 dihybrids with 20,000 genes.
Nope.
Okay, you need more than a little composition notebook.
Yeah.
Okay, so, but what I think is a fair question is to ask, like, how big would the Punnett square be? So if you got 20,000 genes, you could figure out pretty quickly how many gametes they could produce. And then you could figure out the size of your Punnett square.
In theory, you could complete it.
Yeah, so in theory. And you know what? This is a little bit of a thought experiment in computational genetics these days. There is no computer alive that can do that Punnett square. So that's how big the problem is. So in case you were worried I was going to ask you how to do it, not a chance. There is no computer big enough, has enough memory, enough hard disk space, enough anything. There is no computer that can do that. That's how big that experiment would be.
So you'd have about 400 million resulting offspring.
Yeah.
That's crazy.
And think about how many genotypes.
Jeez.
Think about how many different genotypes. Crazy, right?
That's absolutely crazy.
Yeah. So of course, that, to me, that's what makes it interesting. It's fun to have these big, almost intractable problems. So
yeah.
Anybody else? All right. If not, have a great weekend. Looks like it's going to be a little damp today, but I think tomorrow is going to be nice. Big waves this weekend. So if anybody's wandering down to the old jetty down in Holgate, should be big waves. Water is way too cold for me these days. I'm too old. But should be some real nice surf this weekend if any of you guys are into that. So take care of yourselves. Have a good weekend, and look for another email starting Monday. Take care.
Thank
Have
you.
a great weekend.
Thank you.
Thanks.